All right, so I'm going to let Justine kick it off, but we've been really fortunate in my lab to work with Justine and NanoLive on a cool collaboration that we want to get everyone's feedback on.
Yeah, so this is just a little teaser. We're going to talk about some live cell imaging today, as well as the analysis that we can do based on these label-free images that we're able to collect. So thank you to Joelle and Elizabeth. This has been a really fun project to work through for the last, almost a year or so now. So I'm going to give a quick overview of the technology, how our imaging is acquired, and a very brief overview of the analysis, and then hand it over to Elizabeth to do the really cool stuff, which is the real data. So a couple of things. This is just a snapshot of our instrument here. Some things to point out: the top stage incubator here to keep your cells just like they would be in a regular cell culture incubator. While we're talking about label-free imaging today, we do also have a four-channel epifluorescence module, as well as the ability here with our automated platform to image multiple conditions within the same acquisition. Well, we're going to focus my part of the talk on the automated platform over here on the left. The imaging platform that Elizabeth was able to use to collect the images here at the Koch Institute is the manual microscope. The imaging technology works exactly the same way. The only difference really is that automated platform that allows us to do multiple conditions and a slightly different stage to accommodate 4 dishes or a 96-well plate. So if we take a kind of slice through the instrument, the way we acquire our images is through holotomography. So the holography part, where we're measuring the differences in phase shift between the sample beam, as you can see here, where the light passes through the sample at a 45-degree angle. And that sample beam is compared to the reference beam, and we're able to reconstruct the differences there with our beautiful images that you'll see. The second part of the technology is the tomography. So this allows us, with the 360-degree rotation that you're seeing over on the left, to acquire this information from multiple angles. So this increases the resolution, as well as allows us to collect 3D volumetric information within that same single acquisition. So we're using the holography. Is the phase shift of the light is determined by the light passing through the cell and through the structures that have an inherent refractive index property. So what you can see on the left here, if we can play the video again, nope, is, there we go, really quickly here. Structures that have a low refractive index will appear darker, like the media, vacuoles, things like that, where the high refractive index structures will appear very bright, something like a lipid droplet. I'm not sure if you were able to see there, but you can see things like the lipid droplets here, these really bright kind of punctae here. And then the mitochondria, you can see as these long stringies. So all of this is calculated through the inherent refractive index of these structures. So we're able to capture high resolution at an organelle level, as you see here, but in that same acquisition, we're able to collect information at a single cell level as well as a population level by increasing the field of view through our automated platform. It's a super simple workflow and really applicable to so many different kind of research areas. But basically, you plate cells, add a compound or treatment of choice, or different cell lines, or whatever you're interested in. You input your acquisition parameters into the acquisition software, which is very user-friendly. And then it's a walk-away solution. So you press go, and the instrument acquires for the duration that you've input the time lapse that you need to be able to visualize your cellular dynamics. Once that acquisition is complete, it's a one-click analysis, and I'm going to touch very briefly on what our analysis looks like. But one other important point to take away is that these samples are preserved because you haven't labeled them in any way or introduced any phototoxicity. So these samples can be preserved for further downstream analysis as they're still alive and healthy. Always important recent peer-reviewed publications. This is constantly updated and available on our website if you have any questions about specific applications. So while these beautiful images are great and all, how can we take that further and gather data from every well? And not only are you able to run a single analysis, you can always have access to that raw data and be able to run it through any of our digital assays with the same acquisition. So just to kind of lay a baseline of how the analysis kind of starts and how we build our digital assays on this, you're seeing here a single time frame here of a static refractive index image. So this is again label-free. The software automatically segments each cell at each time point, and then we're able to extract a range of metrics for each cell for each time point for the duration of the acquisition. So we extract metrics at a population level, a morphology level, and down to that subcellular content level. And a couple of key takeaways here, because we're using refractive index to generate our images, we're able to measure things like dry mass or and granularity, which is kind of a textural measurement of distribution of organelles within the cell. So we can build on this kind of base analysis and apply this further to specific applications. So we have 3 digital assays currently available. We have our cytotoxicity assay, a T-cell assay, and a smart lipid droplet assay, which is how we actually first started talking to Elizabeth. And I also want to just kind of a point for a smaller company. We're constantly developing and working on new assays based on feedback that we're receiving from researchers in the field. We're also very agile, so if there's a specific application, we want to hear about it. We want to develop new assays to work with, developing fields. So to start just briefly with the cytotoxicity assay, the software segments cells just like we did in that base analysis, and you can see that segmentation mask here, but we then apply a probability and a classification of a cell being apoptotic, necrotic, or living. And within the cytotoxicity assay, well, that's super cool also, we can also introduce fluorescence here as part of the quantification. So you can see on the right, we're segmenting the cells with that segmentation mask and then further separating subpopulations here. So you can see 2 different populations of cells, cancer cells versus non-cancer cells, and for instance, look at different selectivity of a therapeutic or different responses in these various cell populations. Let me skip that slide just for the sake of time. And then, of course, this is where that fluorescence part comes into play, where we can really monitor the responses of different cells in co-culture with that introduction of the fluorescence. And especially relevant to this audience, we can visualize and quantify the dynamics of particle uptake. So here you're seeing those fluorescent particles, and we're able to actually measure the mean intracellular fluorescence to see that uptake of the particles, but you're also seeing that full context of the cellular response to the particle uptake. Just 2 more little assays to get through. Here we're looking at an example of our smart lipid droplet assay, where we're seeing a cellular response in a pancreatic cancer cell line here in response to oleic acid. And so the software segments each cell individually, but then also segments each lipid droplet within each cell. And we extract metrics at that cellular level, at a lipid droplet level, and also a lipid droplet per cell level. So you can really see how cells are kind of adapting to this treatment with oleic acid. Are the lipid droplets gaining mass? Are they gaining in count? And are there different implications for how those lipid dynamics are going within the different cells? And last but not least, the live T-cell assay, where here the software is automatically segmenting target cells from effector cells and generating unique metrics based on the binding, stress, and killing between these 2 subpopulations. So you can see the segmentation mask here, the target cells in blue, the T cells here in red, and where those 2 populations are interacting, the segmentation mask is in yellow. This is again completely label-free. So just to sum up, before I hand it over to Elizabeth, this is a super streamlined workflow that hopefully can save money, time, reduce waste, as it really is a walkaway solution. And you're able to monitor cells label-free over long periods of time, from hours to days to maybe even a week. And the output is this high content, multiplex, multi-parametric 3D data. So with that, I'll hand it over to Elizabeth to show you the really cool stuff.
Yeah, this has been a really fun collaboration. And as Elizabeth, who's a research technician in my lab, is going to walk you through, we had a lot of questions about, would this be worthwhile for people who study nanomedicine? And as a great example of a small company who's willing to work with us, they said, let's give it a shot. That's the laser. Let's go for it back. Yeah, sorry, I didn't. Good.
Okay, so thanks, Justine, for covering all the hefty microscope parts that I cannot cover. But my name is Elizabeth, and I'm an associate in the Straehla Lab. And today I'll just be covering the actual data portion of using the benchtop cell imager. So the story begins with a solute carrier called SLC46A3 that was identified as a potential biologic regulator of nanoparticle uptake in a pooled prism screen conducted by Natalie Bunka and Joel Straehla. It's really only known as a lysosomal membrane transporter, and the gene is what's known as orphaned, so it's not really well characterized in literature. However, in this prism screen, it was identified specifically in the context of uptake for liposomal cores. So we kind of see that when SLC is highly expressed, that there is a decrease in liposomal uptake with less of an influence on PLGA or polystyrene cores. So this led to a characterization in two different model cell lines. There's one cell line with endogenously high expression of SLC called T47D, and LoxMV, which has endogenously low expression of SLC46A3. So then in these two cell line models, isogenic cell lines were created with either SLC overexpressed or SLC knocked out, and then assessed via flow cytometry for bare liposome uptake. And when SLC46A3 is knocked out, we see an increase in uptake of liposomes, whereas when it's overexpressed, there is a significant decrease in the uptake of liposomes. And for the rest of the presentation, I'm just going to say SLC for short. So the identification of SLC as a potentially negative regulator of nanoparticle uptake has led to an ongoing project in the Straehla Lab with the goal of characterizing its function, as well as further understanding the role it might play in nanoparticle uptake. So our current potential hypothesis is that it plays a role in lipid metabolism, and we wanted to characterize this both in vitro and optically with microscopy and in vitro assays. So we started with a BODIPI assay to kind of just assess lipid content in live cells, but the data was a bit messy, and the process was quite time consuming from start to finish between like setup and then image analysis. But with impeccable timing, there was a bio show here at the KI where I met Justine at the NanoLive booth, and they were advertising lipid droplet counting technology, so that was perfect for the project. So we set up two initial demos before the in-house demo, and at the first demonstration, we kind of were like, hey, wait, could this be used for nanoparticles? And then the second demonstration, we had some difficulties with phototoxicity and transporting the cells to their location. So that led to us hosting an in-house demo here at the KI where we were better equipped to kind of have our cells ready and have our nanoparticle suites on hand. Yes. So overall, some of the objectives that we had were to conduct an in-house demo and optimize it for long-term live cell imaging without phototoxicity and with a variety of nanoparticles. Some other kind of side goals that I'll touch on were to see if the refractive index technology could detect lipids from nanoparticles. We also wanted to assess the lipid droplet counting capabilities in our SLC model. And then we wanted to interrogate the analysis software to see if it compares well with the data that we've already collected prior to collaborating with NanoLive. So as Justine mentioned earlier, an important limitation is that we were using the 3D benchtop cell explorer, which is just a single frame in manual focus. which doesn't have the automated capabilities that the 96-well microscope does. So the first nanoparticle I'll talk about is a liposomal chemotherapeutic called Vyxeos. This is a synergistic therapeutic formulated from cytarabine and daunorubicin. Combining the long-term imaging capabilities with the analysis software, we were able to conduct a robust viability assay and kind of see the exact timing of cell death when we use these drugs. So this cell line is Loximbi. It's a semi-adherent melanoma cell line that was initially used in the PRISM screen as well. I'll try and replay the video. So what's pretty cool is that we can see the exact moment that the first cell dies, which is the same in both the control and the SLC overexpressing line. However, then we kind of see the control cells die a little bit more faster than the SLC overexpressing population. The dose that I used was a bit high, so we see all of this happen within 6 hours of imaging. So if repeated in the future, probably a lower dose so we can really expand on this separation. But continuing into the data and the metrics gathered with the EVE software analysis, we are able to generate this plot, which shows the fraction of cell death over time. We can break it down frame by frame and see that the control cells die sooner than the SLC overexpressing cells, suggesting some sort of decrease in uptake in the SLC overexpressing cells. And this is kind of useful because we can monitor the exact moment it happens, whereas in a traditional viability assay, you only have one endpoint readout. And here we can see at an exact point in time what the difference is. So there are some other valuable metrics generated by EVE, like the rate of cell death, the percent of living cells over time, and the percent of dead cells over time. And we still see the same trend hold true that SLC is somehow less sensitive to nanoparticles than the control cells. But for the sake of time, I'm not going to cover all the metrics, but I just wanted to show that the analysis software is generating a variety of metrics for a variety of questions. And these are the ones that answer our questions. And it's important, again, to keep in mind that this is just with the manual microscope, which is N=1. But with the automated one, it would probably be much more suited for high throughput assays. So next are fluorescent nanoparticles. So here I'm assessing lipid nanoparticles with Cy5 fluorophores. These were provided by Namita Nabar from the Hammond Lab with an average size of about 91 nanometers. This particular movie is at a frame rate of 1 fluorescent image every 15 minutes, which is what I determined to be optimal to have no phototoxicity from the laser. And I ran this for about 12 hours, but it gets kind of saturated at 9 hours. And we can, again, see that there is lower signal in the SLC overexpressing than in the control cells. And this is kind of something you can only monitor with live cell imaging, otherwise you'd have a very complicated flow assay. So using the analysis software, we can quantify the fluorescent signal over time. And in this first signal, we are looking at the mean fluorescence background intensity based on area. And it's pretty consistent between the control and the SLC overexpressing, meaning we're not seeing noise. And we can compare these two movies since they were done separately. And then another metric generated is the mean fluorescence intensity per cell, normalized by cell area. And here we see that distinction again, where the SLC overexpressing cells have reduced nanoparticle signal compared to the control cells. So finally, kind of full circle on what we initially approached NanoLive for is the lipid droplet assay. So this was performed pretty easily in 2 20-hour acquisitions, essentially serving as our untreated control. So since I wasn't really using any nanoparticles, I could speed up the image acquisition, and you get some really nice mitosis, very clear stages, which I thought was really cool. And I did use the same laser settings as when I have nanoparticles, just to further validate that I'm not killing any cells just by virtue of having the laser on. And the statistics generated by EVE continually are still pretty robust. And we can even kind of see as the movie progresses where the SLC ones started dividing sooner. So they were higher in confluency, but then eventually they meet back up. This is integral for how we interpret the data, because if we're just blindly comparing them, but they're at different confluence, then we're making improper assumptions. But with these statistics, we can then better interpret the data with more knowledge. So we, again, still find that SLC overexpressing cells do have lower lipid droplet content than control cells. And we can see that based on lipid droplet count and total dry mass of the lipid droplets. Being able to assess the lipid droplets label-free also kind of made this assay very significant to our project, because that was the biggest hurdle at the beginning. And of course, this would still need to be repeated and have some other cell lines in the mix, but it's still quite exciting to literally visualize what we had thought or what we suspected about SLC. So just because of time, I won't cover too many other things, but I wanted to just highlight some other nanoparticles that I was able to image. So I was able to successfully capture bare liposomes of a variety of sizes, all the way down to 100 nanometers. But it's important to say that NanoLive's microscope detection capabilities are limited to 200 nanometers, but we can still see 100 nanometers. And that's kind of what I wanted to try. So these particles were provided by Ivan Pierce from the Hammond Lab and Michael Moran from the Straehla Lab. We are also able to image layered liposomes. So these ones are layered with polyl-arginine and hyaluronic acid. This was taken after 25 hours incubation. And then lastly, a non-lipid nanoparticle, but significant to show the co-dose between nanoparticles of different materials with different fluorophores. Oh, and these particles were synthesized by Juliana Serrano from the Straehla Lab. And so you're probably wondering if any of this was validated, and yes, it was validated with both a plate reader assay and flow cytometry, but I'll just show the flow data for today. So even with flow cytometry, we see the same significant difference between 400 nanometer uptake, 100 nanometer uptake, PLRHA, and the lipid nanoparticles. And I did forget to mention, all of these images I'm showing for these two slides are in the control cells, because the images from the SLC overexpressing cells were not bright enough to put on a PowerPoint. So we still see the same trend in our project. Finally, it's important to mention that all of the analysis thus far were very streamlined by NanoLive software and macro for Fiji. So for each of the movies, they were all processed with the same reference macro to generate a maximum intensity projection movie. And then there's also like a very quick segmentation step, and then the rest of the metrics are generated by Eve. So it gets rid of kind of all that heavy processing. So overall, the things that we wanted to see for our project, when SLC is overexpressed, we see a slower response to liposomal drugs, decreased nanoparticle uptake, and reduced lipid content. This is all very significant in our lab when studying SLC, so that we can find new advantages for nanoparticle delivery. And then in terms of NanoLive's technology, it's quite useful for an alternative approach to viability studies, long-term label-free imaging of live cells, robust and efficient analysis, particularly with their software, and saving lots of time, money, and labor, especially on my part, because I've done these assays with NanoLive technology and with other microscopes, and there is quite a dramatic difference in the workflow. So big thank you to Joelle for letting me pick up a fun side project that turned into something really cool for our lab. Thanks to Namita, Michael, Ivan, Juliana, anyone who provided nanoparticles for me to look at. Thank you to Justine and NanoLive team, who were very flexible and helpful as I learned how to use this microscope. And honorable mention to Jeffrey Kuhn for being a great consultant, as I had many microscopy questions. And thank you to the Marble Center for giving me a chance to talk about what I'm doing upstairs all day. Thank you.